What do you think are the biggest misconceptions about cancer?
Now, more than 50% of patients that have an advanced melanoma, actually they will not die of melanoma. Netrin-1, actually it helps the neuron to survive.
Patrick Mehlen is the CEO of Netris Pharma, a company working on a whole new way of treating cancer by forcing cancer cells to self-destruct.
Researchers are more interested in curing than to find a way to predict that you will have a disease. I said, okay, maybe I should move from a scientist who is interested to understand, from a scientist who is actually interested in serving. So can I do something to help these people?
In this episode, we tour his lab, trace his career, and hear how their new drug aims to flip cancer cells' survival switch back on, reawakening their built-in self-destruct program.
A very scary moment was actually when the first patient received our treatment. I did not sleep because I did not know what would happen. So for me, it was like, it works.
We're in Lyon, inside the lab where a new cancer treatment is taking shape. Patrick Mehlen and his team are developing NP137, a drug designed to switch off cancer's survival signals.
So this is cancer cells that she's growing on the tissue culture line. So you want to see it on the microscope?
I would like to see it. What kind of cancer is this?
This is neuroendocrine tumor, so it's actually a rare cancer. There is not very much study on it. We don't understand too much how it works and how we can treat them.
So this is more a schematic representation. This is the lock, and this would be the ligand, the key.
The key.
Exactly. So, yeah. You see, but it's something related to netrin and receptor.
How many hours you work every week?
I don't work. This is just pleasure.
Well answered. Could you tell us in the most simple way, what is cancer biology and why people develop cancer cells?
So cancer biology, it's a big thing. So basically the idea of cancer is that some cells are becoming crazy. So every cell in our body is actually born, is proliferating. So one cell is becoming two cells, 4 cells, whatever. And then the cells, they are dedicated to one function. And then when they accomplish this function, they die. So this is a classic way of life of a cell. So you have, cells that will live only a couple of days. Like for example, the cell in the intestine, they usually live 2 days, and you have our neurons that basically live for all our life. So it's really depend on the type of cells. Classical view of cancer is that at one point you have some cells that are becoming crazy. Actually not that crazy, more creepy I would say. And somehow they don't want to die, and they want to self-exist. So they will actually proliferate and they will form another organ, actually. So it's like, and it's actually something that we are really looking into it. It's like, a new organism that is developed within our body. And this is basically cancer progression.
Okay, and what caused those cells refuse to die, and why they can bypass that life cycle of themselves to be still alive?
That's a great question. It's actually a great question. We don't have all the answers. So there is some part of it that is linked to genetic alteration. So basically, we have our DNA that encodes for all our genetic information. And so, because of bad luck, because you expose yourself to sun, because you smoke, then you will have specific genetic alteration that will make the cell becoming crazy. So this is one explanation. Other explanation is also that some cells, because they are located in a specific tissue, at a certain moment will actually become crazy, and our immune system, the immune cells, usually they are here to kill these cancer cells. And for reasons that we don't understand completely, so in some cases they don't do that. Actually, in some cases, that's because the cancer cell is becoming very smart and actually produce a system that silence the immune cells. And so the immune cells do not kill the cancer cells, basically.
So they bypass the
Exactly,
immune
it's a way to
cells.
bypass, the normal thing would be that the weird cells should die, and there is what we call apoptosis, so this programmed cell death that is occurring to actually kill this. We do develop a few pre-cancer per day, in our body, but actually we get rid of it. Either the cancer cell by itself is undergoing cell death, or the immune cells, our white cells, are coming here and actually killing them. So in most of the cases, we don't develop cancer, and in some cases, we don't know exactly why. This cancer cell will actually overcome. They will find a way to develop and form a real cancer.
And Patrick, what do you think are the biggest misconceptions that people don't know about cancer?
General misconception is that people are still very afraid of cancer, so somehow everybody is considering that it's still a disease where you will die. And actually this is not true anymore. Because of the research that we've been doing over the last 50 years, we really improve the quality of care, and basically now we cure around 50%, so 50% of patients.
That means they're from later stage or early stage?
So this is one of the points. So usually, of course, it's easier to cure definitely someone who will actually be diagnosed very early. So when we take cancer at early stage, so very often it's your cure and you never heard about your cancer after. But then at later stage, still now we have made huge progresses in we add over the last 10 years different types of cancer therapies like immunotherapy, targeted therapies that completely changed the game, and somehow now you have patients who had very, very advanced cancers that were treated under immunotherapy and basically are completely cured by that. I would take, for example, the example of melanoma. Melanoma 15 years ago, you would be diagnosed with an advanced melanoma, you would die, basically. That would be the sentence. And now more than 60%, 60% of patients that have an advanced melanoma, actually they will not die of melanoma, they will die of something else because of the immunotherapy. So basically this therapy that is here to actually make your immune system work again against cancer. So I told you, initially that some cancer cells escape from the immune cells, and so basically we develop drugs that tells the immune cells to recognize the cancer cell, and so they kill them again. And for example, in melanoma, it works very well. Overall view of cancer changed from a completely, very dark disease to something that is more, you will actually have cancer, we will be treating you, it will be better. And then at one point you will have a relapse, the cancer will come back, and then you will need we will need to treat you again. So it's more a long journey with your disease rather than something that is definitely lethal. So this is the point on general population. And then if we go more to the pharmaceutical industry and to the research on cancer, what I would say is that a lot of the research and drugs that have been developed so far are really focused on the cancer cells that are highly proliferative. When we consider a cancer, we say it's actually growing. And so what we want to do when the cancer is growing is actually is to treat with a chemotherapy, this sort of poison that kills cells, because we think it's good to kill these highly proliferative cells. What I believe, and that lots of people are starting to consider, is that actually this is not the one we should focus on. We should actually focus on these cancer cells that are hiding. So the ones that are not proliferating so much, that are somehow smarter than the other one. And these ones, they don't respond to all the new treatments that we develop, the chemotherapy, immune checkpoint inhibitors, targeted therapies. They all actually target this highly proliferative cells, but not the ones that are hiding. And that are the consequence, the cause of this relapse. You know, when you've heard about this thing where you go, you get a chemotherapy, you have a response, you feel better, and then a few months, a few years later, you come back and you have a relapse. And very often, when you have this relapse, we cannot do much more, and it's because of these cancer cells that are hiding. And this is really what our work have been taking us. And what I think is really the things that we should focus on, generally speaking.
And can you tell us what make you starting to study this subject, and also what is the approach of Netris?
So what I was interested in was on cell death. So I told you that if you every cell in your body is supposed to be born and do something and then die. And then I was interested in the process of dying, because, I mean, even though it sounds dark, die, but actually it's very important, because if you don't die, then it's becoming a mess. The cells need to die in order our organism actually works properly. And for example, if we go back to the brain, 90% of our neuronal cells are dying before we actually get adult. And it's important because it's a way of selection, and if we don't do that, then basically we would not be as smart as we are. And there is many examples. For example, in our intestine, the cells are only living for a couple of days because they're exposed to so many toxics. So if they were living longer, so you would have lots of genetic alteration, and so we would do much more cancer. So these cells are dying after 2 days because they need to die. And if they don't die, then this is where cancer is appearing. So going back to what I did initially was actually to look why normal cells. Initially, I was not doing cancer biology. I was a cell biologist, so I was looking at the cell like here, and I was actually trying to see why cells would be surviving, for living or dying. And we found a sort of weird paradox where we have been showing, so it's a bit complicated, I'm sorry about that, but where basically at the surface of the cell you have sort of lock, so we call it receptor. And basically this receptor, they recognize a key, so imagine a lock and a key, and the key is what we call a ligand. So the ligand is actually something that is produced by other cells, and this key actually comes to the receptor and tells the receptor that everything is okay. So as long as you have the key, the receptor, so the lock is saying, okay, everything is fine, the cell can survive. Oh. And when the key is disappearing, then the receptor say, oh, something is not correct. I should send the message to the cell that the cell should die.
When you observe something a little bit bizarre, what do you feel at that moment, and what makes you feel like, okay, I need to bring this study forward? Exactly.
So for me, because this is my personality, I'm actually more interested in things that are bizarre and things that are, I don't like follow others. I would say that in the world of research, there is lots of researchers that are more follower, and it's good to have followers, so people who would, do things like others, and there is other people who prefer to do things a bit differently. So sometimes it's not easy, because when you do so, the other researchers look at you and say, okay, what are you doing? This is not how it should work. So it's always a bit, it's associated with some negative feedbacks. But then for me, it was actually rewarding. It was a way to be different. Yeah. And so basically, going back to this discovery, so we tried. So it was really basic research initially. Trying to understand how this receptor, this lock, were able to induce cell death, more cell death. And then actually I was invited to start a lab here in this cancer center. So basically, it's a place where we are working on cancer and where patients are actually treated for cancer. And so, coming here every day, looking at the patient that actually had their chemotherapy, that were actually suffering, you have situations that are really, really complicated. I said, okay, maybe I should move from a scientist who is interesting to understand, from a scientist who is actually interested in serving. So can I do something to help these people? And so by coming here, I said, okay, so we've found this system of keys and lock. Can we use it in cancer? Because as I mentioned, cancer cells, they don't want to die. So somehow they should have found a way not to die, either by destroying the lock or by
destroying the key.
Exactly. And so initially, again, during a couple of years, we worked on the lock, and we did lots of work on the lock. And at one point we said, okay, maybe the key is actually something that we should look at. And this key, so one of the keys is called Netris. And it's actually the starting point of Netris Pharma, so our spin-off company. And the idea is that Netris, that is this ligand, so this key, we've been showing that this key is actually upregulated, so it's really produced by cancer cell. In order the cancer cell, like that, the cancer cell always has this key, and so they never die.
Okay.
And so we said, okay, these cancer cells, they produce Netrin-1, so maybe we can actually produce a therapeutic compound, so a molecule that will actually bind Netrin, so that Netrin is taken away, and so the lock will actually not have the key, and so it will trigger the message of the cells that should die. And so this is what we did. We developed a drug that is called a monoclonal antibody, so something that is actually going and bind Netrin and prevent Netrin to bind to the receptor, so to the lock. And by doing so, we've been showing, both in cell culture, in animal models, that it was actually killing cancer cells. What we have been seeing is that actually we don't kill every cancer cell, but we kill more specifically the cancer cells that are the ones that I was mentioning before, the cancer cells that are not proliferating very fast, so the ones that are hiding. So basically the cancer cells that are proliferating a lot, that are making most of the cancer cell, we kill them, but we don't do much. What we really do is that we kill these cells that are not proliferating. And then for us, it was, a great observation, still, at the level of the lab, because it would mean that as there is other drugs that are killing this, highly proliferating, chemotherapy, immunotherapy are really focused on this highly proliferative cell. We said, okay, maybe with our drug, we will actually kill the cells that are not dividing, and that the ones that are the ones that are involved in this relapse, when the cancer is coming back. So maybe we should actually combine our drug with a chemotherapy or with immunotherapy in order we could have a complete effect on the tumor. And this is basically the start of the story of Netris to push into clinical development something that would be associated to the normal treatment, so classic treatments, chemotherapy, immunotherapy. We are actually adding this Netrin antibody.
And will this antibody have any side effect?
No, so this is the beauty of it. So it's that all this system I described it to you is actually taking place during embryonic development. So it's really important during the development of the embryo. But in adults, like you and me, basically we don't have Netrin-1, we never have the lock nor the key. It's only important during embryonic development. And for a reason that we don't completely understand, that actually when a cancer is formed again, it's actually a sort of reformation of an embryo. So actually all the embryonic processes are are coming up again, and so netrin, and so the receptor and the ligand are coming up again, and so with our netrin antibody, we specifically kill the cancer cells, so the cells that express netrin-1, but not the healthy cells. So basically, we've been doing what we call toxicology study in animals, and basically we've never seen any sign of toxicity. And then we did a phase 1, so the trial in human, and again we haven't seen any toxicity. So that's sort of the good part. We seems to have efficacy. So we have been seeing patients that are doing better with our netrin antibody, but this is done without any side effects.
When we hear about the chemotherapy and all the other cancer treatment, the patient actually suffer greatly to get the result. Yeah.
So if we go to this line, it's our idea would be that you kill all these enemies, but you don't kill the ones that are hiding. The ones that are really the ones that you should destroy, because this is the ones that will come back. When you will have killed most of the enemy, you have few of them that will not be dead. And what we are proposing is that with our treatment, we are actually going to kill these ones that will be coming back. So far, it's still a drug that is under clinical assessment. So we have 4 ongoing what we call clinical 2 studies. So basically, in cancer, the development of a drug actually goes from phase 1. So phase 1, you are here to look at a small number of patients and you look at toxicity. So you look at whether your drug has some toxicity. Of course, you are in oncology, so you actually take patients that are very, very sick. Often this is patients that have, where we cannot do much anymore. So we will actually propose them this type of treatment. So phase 1 was done a few years back. So we published this phase 1 data in Nature. So this is a big journal 2 years ago. So basically, again, no side effects, so no toxicity. So we treated like 42 patients, so all types of cancer, very advanced, and we didn't see any sign of toxicity. And we did see some sign of activity, even though, as I mentioned, what we would like to do is to combine, so treat chemotherapy plus our molecule. And so in the phase 1, it was monotherapy, so basically it was only the netrin antibody. And actually, we did see some sign of efficacy. At least, 50% of the patient actually benefited from the treatment, in the sense that the tumor did not progress anymore. Yeah. And then we had 2 patients where we had actually a reduction of the overall lesion. So it was so what we call an objective response. Okay. So it was quite interesting. And what was very interesting also is that we were able to collect biopsies from the patients that were treated with our netrin blocking antibody, and we were able to demonstrate that what we've been seeing in our lab, with cells and with animals, it's actually occurring in patients. So the way we think the netrin antibody is working is really what we hypothesized, a few years back, and so it really means that we should be able to combine with chemotherapy or with immune checkpoint inhibitor or with immunotherapy our netrin blocking antibody. And this is what we are currently doing. So we have, I was mentioning 4 phase 2. So phase 2 is more patient, and basically there is a control arm. So basically patient either receive your treatment or not. And now in our case, patient receive either chemotherapy or immunotherapy, or either the chemotherapy or the immunotherapy plus our drug.
What are the current methods to treat cancer, and what you see the future could lead us to?
This is interesting because currently the fashion is to develop what we call ADC. So this is a way to bring chemotherapy to the tumors. In the past we were developing chemotherapy. So chemotherapy is a poison basically that kills more cancer cells than healthy cells. And so basically you always have a balance between the dose you inject of the poison basically. And so you expect to kill more the cancer cells than the normal cells. And you can do that a few times, but at one point you will have so much toxicity that you cannot give the chemotherapy anymore. So what people have been doing in the last 5 years, they've been developing what we call ADC. So basically, it's a way to bring the chemotherapy to the tumor. And so there is a lot of enthusiasm on that. This being said, for me, this is not that interesting because this is again chemotherapy, and so this is again just a way to kill the 1,000 enemies, but not the ones that are resting, not the ones that are the ones that you should kill. And so basically, this is going back to what we say. You know, it's good to have therapies that will target these very highly proliferative cells. But you should also consider to kill these cells that are not proliferating and that are really the aggressive ones that will make metastases and all this.
As you mentioned, we all have cancer cells in us. And our body is incredibly good at hiding disease until it's too late. How are we going to do the health check?
So this is a question of prevention. That's actually a key aspect. Researchers are more interested in curing than to find a way to predict that you will have a disease. So first thing, we do develop cancer, but most of them are they are stopped before they become actually aggressive. You know, our immune system, our human body is here to eliminate this pre-cancer. Yet we do develop cancer, so it's not rare, but still compared to it's only 1 out of 5. It's already a lot, but it's and then this is really a big question: can we find a way to detect it earlier? And so there is a lot of research on that. You know, where we can now take blood samples and analyze the blood samples. You have various tests that you can do. For example, for colorectal cancer, you can actually do specific tests that will predict whether you can have a colorectal cancer. Prostate cancer, you know, there is a way to detect it just by blood collection. So it's coming, but there is still a lot of work to do. And there is still lots of work to do also on ways to prevent, you know, having a better way of having food. We're in Lyon, so unfortunately we have the best food in France, but so we're probably not good in having healthy food. But that's through food, you know, doing exercise, all these sort of things that are really important also to battle the disease.
So from an expert perspective, what are the things we could do to prevent cancer?
You can do exercise, eat better, don't drink, don't smoke. That's really no smoke, no alcohol. That's some things that will actually prevent, you know, lung cancer. This is still one of the predominant cancer in Europe, in China, in lots of places. And you know, you can just get rid of it just by
smoking.
Just by preventing people from smoking. Liver cancer. So in Asia, you have different types of liver cancers than in Europe. For example, in Europe, you know, most of the liver cancer is due to alcohol consumption. You know, so it's really there is ways to not easily develop cancer. This being said, it's not because you're never gonna smoke, never gonna eat bad things, or that you will not develop cancer. Cancer is also a disease of aging. You know, I know that you are very interested in aging. You know, the idea is that if we would be dying, you know, like at middle age, at 30 years old, we will not have cancer. Basically, there is a very small proportion of people who actually develop cancer before 30 years old. We do have some, and it's a disaster, but most of the people who develop cancer are actually people who are getting older. And the reason why is that because, you know, this genetic alteration I mentioned initially. So basically because cells are proliferating, and so with time, as you're getting older, you repair less, and so you have more genetic alteration, and then you make more cancer. So it's really something that is, I would say, by chance or by non-chance.
How much of cancer depending by genetics, and how much is lifestyle?
That's a complicated question, and it's a complicated it's a dangerous answer because people could actually be scared by that. Because we are talking about probability, we all heard about the story of Angelina Jolie, Angelina Jolie. Sorry, So she was in a family. She is in a family that were predisposed to breast cancer. So she had a genetic alteration, what we call the somatic genetic alteration. So meaning that all the cells have this mutation, and so she has an increased incidence of developing a breast cancer. So indeed, it was actually safer. She had already had kids, so she basically get rid of breast, ovary, all the things that are related to that she could have been developed a cancer. But this is the extreme setting. There is very few people who actually have this mutation in a somatic way. So basically, all cells would have this mutation. So basically, you do have predisposition, but it's only an increase of risk by 1.5, 2 times. So then you have to make a balance between the increased risk and the reality, because it's not because you have 2 times more chance to develop a cancer that you will develop a cancer. So it's a very complicated information, and only I would say the professionals, so the people who are doing genetic counseling could actually advise you really. And my advice would be not to do that, by yourself, because having 1.5 more chance to develop a cancer meaning nothing when you compare to the real life. It's nothing.
What are your studies doing with the drug you discover? Is there any beneficial for our brain?
Based on the work that we've been doing, again, this is cell death, and very naive way to say it. Cancer cells, they don't want to die. Neurodegenerative disease, they die too much. And actually what we've shown in the past is that Netrin 1, again, these things that we are targeting with our drug, is actually more expressed in cancer setting, but is actually lost in brain, where actually it helps the neuron to survive. So we did several analyses looking at whether if you increase the level of netrin, then you would be protecting. So far it was mice, it's mice models, so animal models, and indeed it was working well. Yet the drugs that we develop is really for cancer. You would not consider using it for Alzheimer or for Parkinson.
Yeah.
This is more something that we would do once we actually fully demonstrated that the netrin antibody, the drug, is actually working in cancer. Then we would be open to look at other pathologies. This being said, the netrin antibody seems to be very interesting in other pathologies that are not cancer, and in more specifically, we are very interested in a disease called endometriosis.
So what is that disease?
So endometriosis is a disease of women that is associated with pain. This is the uterus that is actually forming extra lesions. So basically, you have part of the uterus, what we call the endometrium, that goes outside of the uterus and grows. So it's like a pre-cancer. It's actually not cancerous at all. No worries, but it's a lesion that is formed outside of the uterus, and it's very painful. So around 1 out of 10 women are actually affected by that. So it's actually very painful, and so because of the mechanism we've been showing in cancer, we've been seeing that in endometriosis, this is actually exactly the same mechanism. So basically, with our netrin antibody, we can actually affect this lesion and reduce the pain. So the idea, so far, is still only on animal models, but we are planning to have a clinical trial, so a phase 2 clinical trial starting in 2026, so next year, on this disease. That will not be cancer, but endometriosis.
I really see female health as an underserved market at this stage, and they're just.
And it was a disease that nobody talked about it up to a few years ago, because it was women health. It was not a market of interest for the pharma industry, and things are changing. And so for us, it's really interesting to see that our discoveries that was made in cancer could actually be useful for other non-cancer indications.
As a scientist, as someone who's so knowledgeable in the space, what do you see the cells in our brain are different with the cells in our body, or they are same cells?
So the brain is actually made of very different cells, neurons. They don't exist, or they exist, but they are only here to connect with the neuronal system. But there is also lots of cells that are very similar. So you would have immune cells, you would have endothelial cells, the cells that are conducting blood. But the brain is very bizarre in the sense that it's a very restricted area. You have a barrier that is here to prevent most of the things to go into the brain. And it's actually a very important aspect because we do have brain cancer. And actually, this is very bad cancer, glioblastoma, medulloblastoma, all these cancers from the brain. And for cancer researchers, it's actually very complicated because most of the drugs that we develop, they don't go in the brain because they are stopped, or they don't go nicely in the brain. So very often, the drugs that we've been developing for tumors that are not located in the brain do not work in brain tumors. So this is a very interesting ways of development. And actually, we will have a clinical trials that will open on using the netrin antibody for glioblastoma opening next year, because this is a tumor where netrin is really highly expressed, and so we hope we could have benefit here.
Why our brain degenerate? What was causing that imbalance?
Yeah, that's a very good question. So far we don't know much. There is even people who are saying that your intestine is actually the first organ that is important for neurodegenerative disease like Alzheimer. So it's probably more complex than that. But for sure, what is happening is that you have neurons or cells that are supporting neurons that are dying too fast, and this is the main explanation where your brain is degenerating. You have less active neurons, and so you need factors that maintain the survival. So that's where what we did could actually be of interest. And actually, there is my mentor, so the one who trained me when I was a postdoctoral fellow in San Diego, who is actually working on Alzheimer's. So basically he's using the same concept that we have been using in cancer. So basically we want the cancer cell to die, and so we develop a drug in order the cancer cell could die. And what he has been saying is that he's using the same concept in order the neuronal cells do not die. And so he's actually having very impressive data, clinical data on the use of a series of molecules that are promoting survival and balancing the overall setting of your brain, and so have very nice effect on Alzheimer's.
At this stage, what are the things make your eyes spark when you think about it?
Yeah.
Like, what's your most passionate about your research?
Yeah, so if we want to go back to some things that I will always remember is when we started the phase 1, so the clinical trial. You have to imagine that I'm a cell biologist, so initially I'm just doing experiment here at the bench. So I am not a medical doctor. I have no training for that. So we decided to develop this drug. We did not know much, And then at one point we started this phase 1 clinical trial. So patients were included in the trial. So first, very scary moment was actually when the first patient received our treatment. I did not sleep because I did not know what would happen. Of course, you can say that in cell culture or in animals, it's helpful, but in real life, you don't know. So this was a very scary moment.
Well, what are the results?
The result was okay because there were no toxicity, so that was very good. And the second moment is I remember that because this is something that I will remember all my life. This is several patients were treated with the Netrin antibody, and I was traveling. I was supposed to give a lecture in Canada. So I was walking on the street, and then the clinical oncologist who was in charge of the trial actually called me. It was very early on the morning, and he said, okay, we have our first patient that responds. So respond meaning that the tumor was shrinking. And so somehow, they only received the Netrin antibody, so it could not be random. It necessarily means that it works, that something is happening. So maybe it's not going to be sufficient to have a benefit compared to the other treatments that have been developed, but at least it means that it does something and it helps people. So for me, it was like, it works, even though it was really early days. From the dish to patient, exactly. Yeah.
How did you come from that role into an entrepreneur? Because it was a huge company that you're creating now.
We actually initially tried to contact, a big pharmaceutical company, and they were looking at us. Okay, interesting, but come back when you will have clinical data. So very often these big pharmas, they are not so keen on taking risk. They prefer something that is actually working, pay more, but something working than take risk with you. And so I decided, okay, if they don't want to do it, I will do it. And then I took it, and so Mao decided that I will bring it up to the moment we demonstrated it works or not, actually. But we hope it's
you raised
work.
your own money at the beginning too.
So it was our own money from healthy individuals. So it was a way to decide to do that by ourselves. We founded together with my co-founder, that is professor at the university, and basically we founded. It was back in 2000, so something like 15 years ago. Sorry, 15 years ago, in 2010, and but then it actually really started when the first people put money on it. So it's basically 2015. The real start of the company is 2015, and now we are 2025, and it's like 10 years of work where we will actually get the phase 2 data by the end of the year. So basically, we will know whether it was worth it.
Yeah.
We'll see whether we really have a benefit for patients.
From your entrepreneurship skills or background, what are the biggest difficulties you have faced during forming this company?
Actually, I learned a lot. It's really, this is what I say to the researchers that I work with, is that at one point, it's actually every researcher should do that once, just to see how the real work is working, because we are somehow protected as a researcher. We need to find money, we do our research, we publish our research, but if everything is quite simple, I would say we know the rules. When you start a company, then you are in a completely different world. You constantly need to find money, and lots of money. So to do so, you need to interact with various types of people. Some are very interesting, some are not interesting at all. But then you have to live with that, and it's a completely different world. That is interesting also to know, because somehow the innovation we do, when you're a researcher, you have no idea that this world is actually existing.
As a scientist, it might be very difficult for you to start your first round of fundraising.
Exactly, yeah.
And also you need to learn everything about finance, and all around that side to be able to maintain a big company like that?
I should say that we are I'm very lucky to have very good people who are here to help on the financial side, on the business side, on the clinical side. So lots of people are here to help. We have been very lucky also to have very kind investors that have not only put money in the company, but have also been always supportive, So somehow we have been very lucky. I know that this is not the case for everybody. We keep hearing of, companies, biotechs that are, having very hard time because of the investors they had. So far, we've been very lucky. So still, I'm a scientist. What is really of interest for me in my life is actually data. I'm really keen. I still have my lab here. I still love talking about science with other researchers. But somehow this is what I decided to do. I want to push my baby up to the end to also demonstrate that all the research I've been doing is useful.
What is the end, the next step or next milestone look like for you?
The next milestone will probably be, I hope, will be that a larger company will take over and take the drug to the market, because here I will not have too much to add. Again, I'm still a scientist. So at one point, so really what we need is now to bring to finish the clinical trials and to demonstrate that it works, and it works better than the others.
And what are the best advice you receive as an entrepreneur or as a scientist?
For the scientific part, I would say that never listen too much to others. Find your own path and trust what you believe.
What's your driving force to keep going forward?
The things that makes me continue is when you walk, when you enter the building, you have all these people with the chemotherapy that and somehow you know why you're here. You're not just playing around, trying to get money. What you need to do is really to find something to help these people.
Looking forward to 10 years, 20 years, where you see Netris will become?
The beauty of the netrin blocking antibody, so this the NP137 that we call, is that it could actually be used for all cancer type. It's just something that you will add up to every current treatment, or even if you want to consider long term, you can even consider giving it before you have a secondary cancer. So somehow we give you a chemotherapy, we give you an immunotherapy, you are cured. Officially you're cured, but still you have these little guys that are here and hiding. So we can give you the netrin antibody to avoid the cancer to come back. So then it opens a huge possibilities, because basically everybody could benefit from it.
What is your health tricks for our audience if we want to stay vital and healthy with a good health span?
You have to be positive. I would say, positive, eat healthy, do exercise, and stay positive.
Stay positive. Yeah, I think that's very important.
That's very important.
I totally feel you. Thank you for watching the HumanWare Project. To help us to getting more amazing content out there, please subscribe. It means a lot and it will help us to grow. And let's go on this journey together.